today we're going to cover sort of three topics. One is what's called short-term synaptic plasticity. And then we're going to cover a simple learning paradigm to relate plasticity to learning and memory. And then we're going to cover a little bit about the hippocampus and in terms of long-term plasticity. We'll talk more about hippocampus when we talk about memory. Okay, so we'll sort of skip some of the hippocampus levels. So we'll use it as a petri dish for understanding long-term memory today. Okay, so this is another way to kind of look at the system. So short-term plasticity is involved in processing information. And as we look at the mechanisms, we'll start to see why. Longer-term forms of plasticity are ones that are involved in what we consider to be learning and memory. Okay, so these are involved in learning and memory. And then there are other forms that are involved in development and experience-dependent modification. Okay. And I just wanted to make that dichotomy rather clear because we're going to see how the different types of mechanisms are involved in these different processes. So sort of a different mindset for each of these different types of plasticity. In the first case, the phenomenon of short-term plasticity that we'll describe really has to do with the basic dynamics of synaptic transmission. Okay, it's a biochemical process. There are time constants involved, and there are resources that are being used. And so the short-term plasticity really emerges out of those processes. Okay, these longer-term forms of plasticity really have to do with modification. Okay, you're changing channels, you're adding channels, you're phosphorylating things, you're driving transcriptional factors, you're making structural changes. Okay, so there's a distinction between the types of mechanisms that underlie these types of plasticity. Okay, so the first thing we're going to talk about is short-term synaptic plasticity. Okay, this is defined as frequency or activity-dependent change in synaptic amplitude at relatively short time scales. Okay, so there's a real time scale difference here. We're talking about milliseconds to seconds. Okay, this is a time scale in which you're processing information. Okay, it's not the time scale of I need to remember tomorrow what I'm doing. Okay, so this is sort of really has more to do with information processing. Remember all of those sensory processes that we talked about, just because of the time scale involved. Okay, so it's time scale of immediate perception. It's incredibly ubiquitous. It's found at every single synapse to some degree, but there's a lot of heterogeneity in which synapses respond in which way to activity. Can kind of ignore this sort of temporal filter. But it can basically act like a filter at the synapse. And we won't really get into that. But this sort of filtering idea really means that it has functional significance for the neural coding that we talked about earlier. Okay, but that's about as far as we're going to get with that. Okay, so what do I mean by short-term change in synaptic amplitude? So here's a really simple example. On the top, you have the presynaptic activity. So here's an action potential, and then followed by another action potential. This is connected to a postsynaptic cell, and what we're looking at here is the synaptic potential right here. So this is the synaptic response. Note the difference in scale. This goes from -65 to 0. These are overshooting spikes. This goes from -65 to -63. So this is a 2 millivolt EPSP. And then that's the response to the first spike. The second spike, the response is bigger, okay? And it's this change in amplitude is what we really mean by plasticity, that there was a change, and it's related to the proximity of those two spikes in sequence. Okay, ignore these little blips right there. Okay, here's another example. This was an example where the response got larger. Here's an example where the response got smaller. So again, what we're emitting are the spikes in the presynaptic cell, which we could put right up here. And this is the synaptic response. And it's negative going because now we have a current. Before we were talking about voltage. So the previous slide was an EPSP. This is an EPSC. But it doesn't really matter for what we're talking about. you can see that the absolute amplitude is this large for the first one, and it gets smaller for the second one. Okay. Commonly, we define these kinds of changes as the ratio of the second over the first, so A2 over A1, A for amplitude. And you can plot that. And then you can plot that relative amplitude as a ratio, so relative EPSC amplitude. If it was the same, it would be 1. Okay, so 1 means no change, the way we're plotting it here. And if A2 is smaller than A1, then this value is going to be less than 1. Okay, so here a ratio less than 1 is what we call depression. Okay, the amplitude got smaller. So we term that short-term synaptic depression. But the timing of the presynaptic activity really has a big impact on the size of these amplitudes. Okay. So if you have a very short time between the two presynaptic stimuli, the delta t here, you have a rather large decrement in the amplitude. And then as that time difference gets larger, so you have a longer time between those two stimuli, this essentially goes to baseline. So if you wait long enough, you have the same amplitude again and again. Yes?
I'm sorry, what does EPSC stand for?
Oh, EPSC, excitatory postsynaptic current.
Okay, current and that is
Current.
potential.
Right, so that's what you measure when you're in voltage clamp. You measure EPSC. In current clamp, you're measuring voltage and you get an EPSP.
Thanks, sorry about that.
Okay. Good to clarify. Okay, so as we go along, we're going to be basically pulling from a lot of our past lectures, and I'll try to point out a couple of specific spots. So the opposite of short-term synaptic depression is short-term synaptic facilitation, meaning the amplitude gets bigger with activity. Okay, so here's an example. Here's an EPSC1. EPSC2 is significantly bigger. And just to confuse the issue, these are being plotted slightly differently. But just notice that the amplitude is big, and then it declines down to a baseline. So here's 0. This is a difference measure. So here 0 means no difference. So if the amplitude is the same for A1 and A2, that would be 0 in this plot. Okay. But you can see again that if the time difference is small, you get the largest differences, the largest increase, and then that increase declines over time. Okay. And this sort of effect is thought to be related to the theory that there's leftover calcium in the presynaptic terminal. Okay, and we'll sort of summarize this in a minute. But to analyze this, you can measure the calcium fluorescence in the presynaptic terminal, and you can measure the time course of the calcium fluorescence. So initially, there's a big increase when you have a synaptic input, and then it decays. Okay, and this is the calcium rushing into the presynaptic terminal, which of course causes release. But then it takes time to sequester that calcium again. Okay, and while that is going on, if you have another stimulus, say right here, the calcium levels are rather high. And we all know that calcium exocytosis depends on calcium levels. So if the calcium levels were already high, then you're going to boost the response. Okay, and that's what causes this increase in the amplitude. And as that calcium declines because it's being sequestered by cellular mechanisms in the terminal, the amplitude declines. Okay, so maybe that's a coincidence. So you could do a little manipulation. So remember, we can apply a calcium chelator to the presynaptic terminal. In this case, the calcium chelator allows an initial influx, but then rapidly binds all of the calcium. Okay, so you have that initial influx that causes exocytosis, but then you've got these molecules in your presynaptic terminal which is binding all of the calcium. Okay, so the signal here, this residual calcium signal, is truncated. And what happens is that the facilitation also gets truncated. So here it follows the time course of the calcium. Here it also follows the time course of calcium by being shorter than in that case. Okay? So this is supported that the degree or the time course of calcium in the presynaptic terminal is important for this aspect of plasticity. Okay, and so here's basically you should realize this is a review. Go back to lecture 6 and review the process of synaptic transmission, all the steps in chemical transmission. But we can relate this plasticity directly to those steps. Okay, so here we have the action potential, which causes voltage-gated calcium channels to influx the calcium. The calcium causes synaptic vesicles to exocytose, releases a neurotransmitter, which binds to the postsynaptic receptors. Now, as you use these vesicles, you have to replace them. Okay? We talked a little bit about that whole process of docking and priming and endocytosis on the other end. Okay? And this is a process, and it takes time. And so as you use vesicles, there's a process with a particular time constant that replaces those vesicles. And if that process is slower than you're using them up, you can, what we call, deplete the vesicles. So you can have fewer vesicles available. And that is part of what causes synaptic depression. Another possible source of synaptic depression is that calcium itself can inhibit the calcium channels. Okay, that's another potential. The facilitation we talked already is due to the residual calcium. And the reason the residual calcium is so important is because every one of these vesicles has a release probability. Okay? So I want you to remember this term, release probability, or probability of release. Okay? So those vesicles are sitting there, and they're what we call stochastic. Okay? When you have a calcium influx, they may or may not release. And the likelihood of their release is what we call release probability. Okay? And it depends on lots of different factors. But it could be anywhere from close to 0, meaning they're unlikely to release, to close to 1, meaning they will release if they see calcium. Okay? And usually it's somewhere in between. Okay? And the more calcium you have, you can modify that release probability. So you can up your chances of releasing a vesicle by just having more calcium around. Okay. And often we abbreviate this PR, release probabilities. Okay. And different synapses basically have different baseline levels of release probability. And this influences what kind of plasticity you're going to see at these synapses. So on the one hand, you have a synapse here that has a very high P, okay, very high release probability, and it has a tendency to depress, okay, and that's because high release probability means you're going to use up vesicles quickly, and you may or may not have time to replace them. Okay. On the other hand, if you have a terminal, synaptic terminal here that has a relatively low P, okay, your initial response may be smaller, but it has more room to increase its release probability. You start sort of at 0.1, and you have residual calcium, and you go up to 0.3. That's like 3 times the release probability, and you're going to get a larger response. Does that make sense to everybody? Okay. Yeah. So. Okay. Does anybody have any questions on this short-term plasticity? Yeah.
Is high P and low P, is that probability?
P is probability, yes. So in this case, high P is the high release probability, and low P is the low release probability. All right. So let's see if we can sort of use these in a circuit. Okay, and we're going to talk about a basic form of learning that some of you have probably heard of before. We're going to talk about, and these are sort of somewhat specialized terms that I'm going to use for this particular system. The plasticity literature is a little confusing because people use terms slightly differently. Okay, so this is the definitions that we're going to use in this system. Habituation is going to be a behavioral, I should probably write this, behavioral decrease in response to repeated stimulus presentation. Whereas sensitization is going to be generalization of an aversive stimulus to non-aversive stimuli. Okay. let me just sort of write down. Okay. Habituation and sensitization. In this context, we mean this in a behavioral way. Okay. We haven't talked a lot about behavior. We're going to start to talk more about behavior, and we'll see. Okay, so Aplysia californica is your humble little sea slug that has had a big impact on neuroscience. Okay, so here's your little guy. And the important parts of its anatomy is this siphon and gill, and this is known as the mantle. So here's the head of the animal, and here's the tail of the animal. So a very simple invertebrate that shows simple forms of learning. I would think that is such a simple animal would be able to learn, but it has very rudimentary forms of learning, and we can learn a lot from it. Okay, so the first form of learning that we're going to talk about is what we call habituation of the gill withdrawal reflex. So let's take a minute to actually look at this. Let's go back for a second. Let's talk about habituation first. Okay, so if you touch the siphon, okay, you use a little probe and you touch the siphon, the animal will respond by withdrawing its gill. And I'll show you a video of what this looks like. But it'll have a behavioral response to touching its gill. Okay, so here's the first touch. Okay, and this is magnitude of gill contraction, so it withdraws it a lot. Okay, if you keep touching it, keep poking the animal. Eventually it'll go, meh, doesn't care anymore. It realizes you're not a threat, and the amplitude of the response goes down. Okay, so this decrement of the response is what we're calling habituation. Okay, if instead you give it a strong aversive stimulus, which is a shock to the tail, okay, it will give you so if you give a shock to the tail and touch the siphon, it will give you a larger response. It'll basically recover a lot of this response now. So it did habituate, and now because you shocked it, it's going to respond a lot to that tail, the siphon touch. Okay. Now we'll look at the response.
Extremely weak stimulus, so you can see the amplification with sensitization. Weak stimulus to the siphon, you see a modest withdrawal of the gill.
Can you see it there?
Now we're going to frighten the animal, startle it, with a noxious stimulus to the tail. That, of course, causes a contraction in its own right. This contraction lasts for seconds, but you can come back minutes later. The same siphon stimulus now produces a much more powerful withdrawal. We can now compare the two, normal and sensitized, and see how much more powerful the withdrawal is in the animal that is stopped. The memory for this event is a function of number of training trials. So if you give a single tail shock, plotting here, change in reflex strength is a function of time. The enhancement of the reflex lasts minutes. But if we give the same stimulus 4 or 5 times, we get a memory that lasts a couple of days. So we have a short-term memory and a long-term memory, and we can now look at the difference between them.
So, exciting as it is. Okay, so that was Eric Kandel talking about this tail shock. So I wanted to point out that this particular panel is a little confusing because the shocking and the touching of the tail occurs sort of before this trial, and then this is in response to just the siphon touch. So obviously if you just shock it, it responds really strongly because that's a strongly aversive stimuli. This is the response to a siphon touch after you've paired the tail shock. Okay, so that's a little clear. So this is a plot of this gill withdrawal response. So now 100% is the normal response. And when you don't have a shock, you see that this response goes down. That's habituation. If somewhere in the middle here you give it a single tail shock, and then give it a siphon touch, you have this rapid increase. In fact, it's more than it was before, even just with a normal siphon. This is the sensitization, but then it eventually goes away. Okay, and that's with just a single tail shock. And now if you pair the siphon tail shock multiple times, you can get longer forms of memory. Okay, and this is where we start to get into sort of genuine learning, genuine memory. So let's see. This is a 4 single tail shocks, and this is in hours. This is in days. So even days, a day later, you get a response, and then it kind of goes away. If you do 4 trains of tail shocks, you can get a response that lasts a little longer. If you train the animal on 4 trains a day for 4 days, you can get much longer learning. So the more you train the animal, the more you can engender this memory.
Train.
Okay, so that's all very interesting. But the real key to this system and why we're teaching it as a neuroscience, fundamental part of neuroscience, is because you can really get at the circuits that underlie this simple behavior. Because the aplysia as an invertebrate is a relatively simple system. This is the ganglion of the aplysia californica, just aplysia. And each of these are cell bodies of neurons. Notice that these are giant. This is 100 microns. Okay, so this is like 100 microns across. You could actually take an electrode and kind of, by hand, record from these cells. In fact, they used to have a little competition at Woods Hole. People would get together and drink beer and see who could hold their recording for the longest time. But basically, you could really access these cells really well. Here's a real example, so you can really see these under the microscope. And this is just a dissecting microscope. This is nothing fancy. Okay. And of course, people can define which neurons are which. And they came up with the basic idea of what the basic neuronal circuit is that underlies these responses. Okay. And so basically, this is the circuit. You have a sensory neuron that detects touch on the siphon skin, okay, and it projects directly to a motor neuron that causes gill withdrawal. Simple circuit, right? Very reflexive. Okay, but obviously it has some complex behavior going on here. Okay, because when you continuously touch the siphon skin, eventually there's less gill withdrawal. So we know something has to be going on in this contact. The second part of the circuit is the tail. So here's a sensory neuron in the tail that receives the tail shock. It contacts the system via an intermediary neuron, this modulatory neuron. Okay, so pay attention to this yellow bit. All right, so we can record from each of these circuit components. So here we can record an action potential in the sensory neuron. So every time you tap the siphon, you get an action potential. And then you can record in the motor neuron. Okay, and you record a decrement in the EPSP. So here it's fairly big. Okay, and here it's smaller. Okay, so there's very clear depression going on at the sensory neuron to motor neuron synapse. Okay. Now when you do the sensitization, okay, which is this little red line here, you see that you get a recovery of this EPSP. Okay. So what that means is that the sensitization, even though it's coming from a different input, is having an effect on this synapse. Right? It's not contacting the motor neuron through another pathway. Okay? It's affecting this synapse. Okay? And it's doing that by releasing serotonin onto the presynaptic terminals. Okay, so let's focus in on this area right here. So this modulatory interneuron releases serotonin onto this presynaptic terminal that comes from the sensory neuron to the motor neuron. I'm going to kind of skip this. This is just an explanation of what those mechanisms that underlie that presynaptic depression might be. Okay, it could be the depletion of vesicles we talked about, or it could be inactivation of calcium channels. Kind of doesn't matter so much for our purposes. Okay. And I want to focus on this interaction right here. So this modulatory interneuron has a facilitatory effect on a second synapse. Okay? And this is what we would call heterosynaptic. It's one synapse influencing another. Okay, so how does this happen? So the biochemical pathways have been pretty well worked out for how this happens. Okay, so here in blue is your sensory neuron. Here are the vesicles releasing neurotransmitter onto the motor neuron. Okay, this happens to be glutamate. This interneuron, this modulatory interneuron, has vesicles that release onto the presynaptic terminal. Okay, notice it's releasing on the presynaptic terminal, not onto the motor neuron. Okay, and this serotonin is binding to a serotonin receptor, and it's triggering this second messenger cascade. Okay, so let's follow along. So it's signaling the serotonin receptor through a G protein. It's activating adenylyl cyclase, which increases cyclic AMP. So in this particular system, that cyclic AMP is going to activate protein kinase A. So it's going to separate the there's sort of two subunits. There's catalytic and regulatory subunits. And it's going to liberate the regulatory subunits and the catalytic subunits. The catalytic subunits is going to modify a potassium channel. Okay. So the effect on the potassium channel is shown here. So here's our control action potential. So this is modifying the potassium channel in the presynaptic terminal of the sensory neuron is going to have an effect on the action potential on the presynaptic side. Okay, so here's the control action potential. Okay, and if we have activation of pKa, the end effect is to basically inhibit those potassium channels. pKa creates a broadening of the action potential, okay? And it does it by increasing the threshold for activation of the potassium channels. Okay. So that's not so important, but the basic idea is that it takes a lot more to activate those potassium channels, and you know if you don't activate the potassium channels, the spikes are going to get longer. Okay. Remember, go back to action potentials, where we had spike goes up, and part of what causes the repolarization is the activation of potassium channels that pulls the membrane back down. Okay? If you modify those potassium channels, it's going to make the action potentials longer. Okay, making those action potentials longer means that you're going to get more neurotransmitter release. You're going to get more calcium influx because the terminal is going to be depolarized for longer. You're going to open the calcium channels for longer, you're going to get more calcium influx, you're going to get more neurotransmitter release. Okay, so let's kind of put this, yes?
What, is it known what exact modification occurs in the potassium channel?
Probably, I don't know off the top of my head. Yeah. There's a lot of potassium channels, and there's a lot of options for modifying it. Yeah. Okay, so let's sort of put this together with a particular rule. So these short-term changes in synaptic strength. Okay, this is what happens immediately after the tail shock. Are mediated by what we're going to call post-translational modifications. And that just means that we don't need to synthesize new proteins. Okay? We're just using a second messenger system to make a modification that has kind of a short lifetime. Okay. So what have we seen happen? We saw depletion of neurotransmitter during the synaptic depression for the habituation component. And we're seeing the facilitation of neurotransmitter release, as in for sensitization. Okay? Okay, but what about those longer term changes? What's going on with those? And it turns out that longer term changes require protein synthesis. Okay, so how do you get that? Okay, so longer-term changes in this system are also driven by cyclic AMP. Okay, so you have activation of Krebs. Remember we saw this previously in the signal lecture 8, I think, lecture 7. So Krebs is a transcription factor. Okay, and it controls a number of other genes. So we have activation of Krebs, and in particular, Krebs will activate a protein called ubiquitin hydrolase. And what happens is that you have a degradation of the, let's see, that's right. Okay, you're going to get a degradation of the pKa regulatory subunit. Back up. Right? So we said the pKa has two components, a catalytic subunit and the regulatory subunit. Okay. So if you degrade the regulatory subunit, that leaves that catalytic subunit loose in the presynaptic terminal. Okay. And that's going to cause a maintenance of those changes in the potassium channels. Okay, so here's our second goal. These long-term changes in synaptic strength are mediated by protein synthesis, okay, via those transcription factors. Okay, so in this case, repetitive activation, repetitive heterosynaptic stimulation stimulates the synthesis of a protein called ubiquitin hydrolase, which causes degradation of the pKa regulatory subunit, which causes prolonged activation of prolonged broadening of the spike through inhibition of the potassium channel. Okay. And I drew a little circuit diagram to see if this would help a little bit. Okay, so serotonin causes an increase in cyclic AMP that causes an increase in the activity of the catalytic pKa, which causes a decrease in the potassium channel activation, which causes spike broadening. Okay, and it does this through phosphorylation. The catalytic pKa also activates Krebs, which is a transcription factor, causing protein synthesis of ubiquitin, which degrades regulatory pKa, which enhances the catalytic pKa. here's your short-term changes, and then here's the pathway that allows those short-term changes to persist over time. Okay? And you only get this activation of this part of the pathway when you've got a lot of activation of the original stimulus. All right, so kind of cool system. All right, so this was discovered by Eric Kandel, who won the Nobel Prize for work in the sea slug for neuroscience, physiology, and medicine. Okay, and this is why the sea slug is so famous. You know, it was really sort of a early way to try and get at memory. You know, Eric Kandel wasn't interested in sea slugs; he was interested in memory and learning. And it was really quite accepted at the time. Everybody thought, okay, there has to be some sort of modification of synaptic connections. But nobody really had a way to get a handle on it. And his stroke of insight was to use a very simple animal to start to get at the fundamental cellular processes that are involved in this. Okay, and he wrote kind of a nice little book about his work. Okay, and you know, really everything that we have learned about the processes that go on in aplasia really apply to our brains as well. Okay, so this is just to kind of collect together some of the terminology that is used related to plasticity. So we already used the word heterosynaptic plasticity. This is the activity of one synapse regulating the strength of another synapse. Homosynaptic plasticity, so same synapse plasticity. Activity of a synapse regulates its own strength. So the sort of short-term depression that we looked at in habituation would be an example of homosynaptic plasticity. You're not involving other synapses. Okay. There can be presynaptic changes. Okay, changes in the amount of neurotransmitter release. So modification using the spike broadening would be an example of a presynaptic modification. And then there are also potential for postsynaptic changes. Okay, we haven't really talked about it in this system, but as we go forward, we're going to talk more and more about postsynaptic plasticity changes. Okay, so next we're going to talk about the hippocampus. Okay, and I want to sort of make just a slight distinguishing comment about the idea that learning and memory we sort of use these interchangeably, but you can think of learning sort of more broadly, perhaps, as a change in behavior over trials. And we'll see in the next in a couple of lectures that you can learn without even know that you're learning. You can be unaware that you're learning. Memory, on the other hand, we used to refer to specific things remembered. So this is more of a conscious perceptive thing. And of course, these are not completely, mutually exclusive. Okay. So in 1949, people have been thinking about learning and memory for a long time, ever since Pavlov's dogs, and trying to come up with a theory of learning. And so one of the early theories that has been enormously successful and has been seen over and over again is what's known as Hebb's postulate. Okay, and we'll refer to different kinds of learning as Hebbian learning. And he's trying to really explain associative conditioning. Why is it that if you pair, a bell with food in dogs, eventually they'll salivate just to the bell? There must be something going on that connects those two things in the brain. And so he said, let us assume that the persistence or repetition of a reverberatory activity tends to induce lasting cellular changes. Okay, with axon A, so reverberatory, he's really kind of talking about spiking activity. When axon A is near enough to excite cell B, and repeatedly and persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells, such that A's efficacy as one of the cells firing B is increased. All right, what does that mean? Draw a little diagram. A, contacting cell B. Okay, and the basic idea is that if you get an action potential in cell A and an action potential in cell B, and you do it over and over again, that you're going to get an increase in the effect of cell A on cell B. Sort of really simple. The other way that people often say this is that neurons that fire together wire together. Okay, we'll see this a lot in experience-dependent plasticity. Okay, but it turns out that this is, really, really quite true of sort of the most fundamental aspect of plasticity that we're going to talk about now, which is the long-term potentiation in the hippocampus. Okay, and we're going to talk about the rodent hippocampus. Right? We took a lot of pains earlier in the course to learn the human anatomy. The rodent looks quite different, so don't be sort of confused or misled. So here's the hippocampus in a rodent brain. And if you take this out and you cut a nice slice through it, you get something that looks roughly like this. Okay, and this is known as the classic tri-synaptic circuit of the hippocampus. So remember, the hippocampus is one of those three-layered cortical areas. Okay, the center layer made up of layers of the cell bodies of the pyramidal cells. Okay, and there's like a streak right here and another streak right here. So the fundamental input is from entorhinal cortex that contacts granule cells of the dentate gyrus. Those in turn contact pyramidal cells of CA3, and those in turn contact the pyramidal cells of CA1. Okay, so DG, CA3 to CA1. This projection from CA3 to CA1 is referred to as Schaffer collaterals. Probably named after a guy named Schaffer, I don't remember. Okay, but this is sort of the fundamental part of the circuit that we're going to talk about, is the CA3 to CA1 Schaffer collaterals. Here's another view. This is a coronal section through the rat, and it has this sort of classical shape of the dentate gyrus and CA3 to CA1 on both sides. Okay, so we're going to look at this sort of a simple part of the circuit. Okay, so you have this CA1 neuron kind of sitting in its layer, and it's sending an apical dendrite up, and it's receiving inputs from lots of fibers. Okay. And if we're talented electrophysiologists, we can use our stimulating electrodes, and we can stimulate independently a little group of fibers that are separated. Okay, so we can differentiate between the inputs, okay, between these two sets of collaterals that go to the pyramidal cell and CA1. Okay, and this is often referred to as, a two-pathway experiment. It's not really two pathways, it's just two separate sets of axons. Okay, there are other setups that use genuinely two different pathways, but separate axons. Okay, so we're going to use a stimulus that is referred to as a tetanus. So a tetanus is really just a high-frequency stimulus. High frequency, short, okay, gets the job done. Okay, so each of these are EPSPs recorded in the same postsynaptic pyramidal cell, okay, before and after the tetanus. So you're going to stimulate one pathway, okay, and record an EPSP, so this bottom one here. You're going to stimulate the other pathway and record an EPSP to that pathway. Okay. So both of these elicit an EPSP in the postsynaptic cell. They're different EPSPs. It's not the same one. They both elicited EPSP. Now, if you use a tetanus only in path 1, okay, so this electrode, you're going to apply a tetanus. The other one's quiet. And then use a single stimulus again to test the size of the EPSP. You'll find that in pathway 1, you have an increase in the EPSP, but in pathway 2, you have no increase. Okay. So 2 things. The tetanus increased the size of the EPSP, but it only increased the size of the EPSP in the pathway that was tetanized. Okay, so it's specific. Now, this modification, by definition, goes on for a fairly long period of time. So here we're plotting the EPSP amplitude. So prior to the tetanus, which is at time 0, okay, we have some baseline level. So these are the EPSPs that you record prior to the tetanus. After applying the tetanus to pathway 1, the EPSP response to in pathway 1 increases and remains high, and pathway 2 stays the same. And it's this length of this effect that denotes long-term potentiation. Most of you will know that an hour is not really long-term in the life of an animal, but further experiments have shown using in vivo preparations that this can actually last for days or weeks or months. Okay. So it is truly long-term, but in the context of a slice preparation, an hour is pretty long. Okay. And that's sort of our definition. Okay, so what have we learned? We learned that we have a very specific increase in the strength of the synapse along the pathway that's been activated, and not along the pathway that's not active. Okay, and this is known as specificity. Okay, and this is incredibly important, right? You want to strengthen the synapses of the inputs that are causing the activity, okay, and that are carrying the activity. You don't want to increase the synapses across the board. Okay. Presumably, each of these inputs are bringing in some different form of information. Okay, so you want to be specific. All right, so how does this happen? Okay, why do you get this kind of specificity? And it's really a combination of coordinating the pre and the postsynaptic cells. Okay. And so let's use a different stimulus. Instead of using this big, strong tetanus stimulus, let's evoke an EPSP in one pathway and do what we think might be causing a change in the postsynaptic cell. So what do you think happens in the postsynaptic cell when you stimulate the presynaptic really hard? Gets excitatory input. Do you think maybe it fires? Do you think it depolarizes, hyperpolarizes? What do you think is happening in the postsynaptic cell when you hit those inputs really hard? Probably depolarizing, right? Okay. So what if we replace the depolarization that's happening with tetanus with our own version of it? Okay. So now we're going to record from the postsynaptic cell while we're stimulating the presynaptic. And instead of using that really strong tetanus stimulus, we'll use just sort of a mild stimulus, like we did with all of the baseline responses. So we're going to stimulate this enough to elicit an EPSP, okay, neurotransmitter release. But we're going to directly depolarize the postsynaptic neuron. Okay? And lo and behold, you'll find that you can get a strengthening of the synapse simply by pairing depolarizing pulses and EPSPs. Okay, so what can account for this? What kind of cellular property can account for detecting when you have both presynaptic glutamate release? Oh, I should probably go back just for a second. Okay, what's not shown here is that you can do this, and there's also no effect on the second pathway either. Okay, so just depolarizing the postsynaptic neuron is not enough to strengthen the synapse in the pathway that we're not also activating with the EPSP. Okay, so it's still specific. So what kind of properties can account for this kind of response? So you have to detect both of these things. You need presynaptic glutamate release and postsynaptic depolarization. And if you remember from lecture 7, we talked about a particular set of glutamate receptors. In particular, we talked about the NMDA receptor. Okay, does anybody remember what was special about the NMDA receptor as opposed to the other one, the AMPA receptor? Yeah.
Magnesium block.
Magnesium block. When does the magnesium block?
When there's depolarization. Oh, when there's not. Magnesium is there except when it's depolarized.
Magnesium is blocking except when it's depolarized. So depolarizing the cell removes the magnesium block. Okay? And so this is what makes the NMDA receptor essentially a coincidence detector. Okay? Because you have to have glutamate first to activate the NMDA receptor. And the cell has to be depolarized enough to get rid of that magnesium block and allow ion flux. Okay? So go back to lecture 7 and review these properties. Okay? But this is what makes the NMDA receptor a coincidence detector. Okay, so what's happening here? Let's look at this in sort of graph form. So when you have an action potential invade the presynaptic terminal, and you have exocytosis, okay, and you have neurotransmitter, and the neurotransmitter is going to bind to each of these two types of glutamate receptors, the AMPA receptors and the NMDA receptors. Now remember, the AMPA receptors have no magnesium block. So if they open, they're going to flux ions, they're going to depolarize. So these allow for depolarization. If the depolarization is sufficient, either because you get so much current flowing in here, or because the neuron is activated generally, it's firing action potentials, or it's just very depolarized, then you're going to remove the magnesium that's sort of sitting here in the channel, and now you're going to have an influx of cations, including calcium. So that's the second thing. So NMDA receptors flux calcium, whereas typically AMPA receptors do not. Okay, so the NMDA receptor is responsible for the calcium influx through synaptic receptors. So you get calcium flowing in. Okay? And it's this calcium signal that's ultimately responsible for the LTP. Okay, so this is known as the dual activation requirement for LTP. You need two things. You need the presence of glutamate, okay, that binds to those receptors, and you need postsynaptic depolarization to remove the magnesium block. Okay, if you have one and not the other, you're not going to get LTP. Okay, so let's look a little bit more closely at this. Okay, so a lot of what we know about LTP deals with neurons that have spines. So we're going to talk a little bit about spines. Okay, so here's a dendrite, the shaft of the dendrite, and then sticking off of the dendrite are all these little protrusions that are known as spines. And in the hippocampus, this is the primary site for excitatory synapses to contact the cells. Okay, we won't talk too much about this, but oftentimes inhibitory synapses contact on the shaft of the dendrite and not on the spines. Okay, so spines are kind of special. And if you have fancy imaging equipment, you can actually look at activity at the level of the spine. So this is an example of that. So here's this dendrite, and there's a little spine right here. There's a little spine right here. You can't even see the neck of the spine here. So this is baseline. And when you stimulate, okay, either spritz on neurotransmitter or stimulate a synaptic input, you see a little spot here that tells you that you have activity. So this is a sort of a form of calcium imaging. And they're talking about a particular calcium protein called CAMP2. Okay, and then after stimulation, the calcium levels go back down to close to baseline levels. Okay. So the NMDA receptor is really responsible for this calcium signal required for the induction of LTP. Okay. To start the process, you need this calcium signal. Okay, so what actually underlies that increase in the EPSP? the 90s, in my memory, there was a big argument. Some people said it was a presynaptic change, and some people said it was a postsynaptic change. A lot about this. It was very entertaining. But in the end, the postsynaptic people won. Okay, and so we'll talk about why we know that. And one of the big clues to understanding how that comes about is through studies of something that was kind of arcane at the time. And it was this thing called silent synapses. And this was something that was studied mostly in development. Okay, but you can so you can observe plasticity. You can observe these long-term changes in the spines themselves. So here's an example of a dendrite with a spine, another little sort of skinny proto-spine. Before LTP and after LTP, if you look at the exact same spot, you can see an increase in the spine head and an actual spine, full spine right here. So you can see these things at a structural level, okay, not just physiological. And so people thought, oh, there must be something happening at the postsynaptic side. You know, what is causing this increase of the size of these spines? And so a big clue to this came from this study of silent synapses. Okay. So silent synapses were a discovery where people were looking for synapses between cells. And they weren't finding them. They were like, we know these look connected. If you label them and you look at how things, you know, intertwine around each other, they look connected, and yet there's no physiological response when you stimulate one and record the other. Why is that? And so finally somebody got the bright idea to do an experiment that looks like this. So typically under voltage clamp, you know, you hold at sort of the standard voltage, -65, close to resting, very normal. And you would cause a stimulus during this. So this is the representation of the voltage that you're holding in the postsynaptic cell, and this little arrow is you're zapping this presynaptic element to get a response. Okay, and so normally you would do this. So this is just the artifact of the stimulus, and then you look and you see nothing. Okay, so it looks like these cells are not connected. But if you step the voltage up to a rather high depolarized voltage, in this case +55, and stimulate again, suddenly you get a big response. Okay, this is an outward current, right? We're in voltage clamp, this is outward, and that's because we're holding the voltage on the other side of the reverse potential. Okay, but the key here is that you're getting a big synaptic response. So, aha, there really is a synapse here. Okay, and the idea was that there were no AMPA receptors. So a silent synapse is a synapse that only has NMDA receptors. And remember what we said about the NMDA receptors in magnesium block? When it's hyperpolarized, the magnesium is blocking. Okay. And that's what's happening here. Is the magnesium is preventing any flux through the synaptic receptors because there's no AMPA receptors here. Usually there's some combination depending on how much the magnesium block. But here, we're so hyperpolarized that there's no flux. Okay. And if you look at this using immunohistochemistry, so now we're using antibodies to label the AMPA receptors and NMDA receptors. So the AMPA here are the red. So you see a lot of AMPA in the shaft, but also in the in some of the spines right here. Then you label the NMDA receptors, and you can see lots of little green dots. Okay, and those are the spines that are expressing only the NMDA receptors and not the AMPA receptors. And those are your silent synapses. Okay, so what's happening here? The idea is that during plasticity or during LTP, that you're taking a spine that originally has only NMDA receptors, okay, and you're adding AMPA receptors. Okay, so during development, what happens is that you have silent synapses that are being transformed into functional synapses by the insertion of AMPA receptors. Okay, so now you have both of those in these mature synapses. whole series of experiments. We now understand that this is what's happening during LTP plasticity as well. That the original synapse maybe has some combination, as a normal mature synapse has a combination of AMPA and NMDA receptors. So that when you have sufficient activation, you get an unblocking of the NMDA receptor and a calcium influx. This calcium signal acts through second messenger pathways that can involve chem kinase 2, but also perhaps protein kinase C. And through a series of steps, you get insertion of additional AMPA receptors. Okay? So you go from, say, 1 AMPA receptor here to 3. Those numbers are probably completely made up, but you have an additional AMPA receptors. And that's what makes the EPSP bigger. Okay? So one implication of this is that the size of that EPSP is limited by the number of AMPA receptors in response, in the postsynaptic side. You might also notice that inserting these AMPA receptors, they're sitting in this little sort of vesicle-like object, and when those are inserted, you have more membrane. Okay? And so that may account for the fact that these spine heads actually get bigger. So that's a structural correlate to the insertion of these receptors. And if you do immunohistochemistry, you can see larger structures in the postsynaptic density here. Okay, so that's the induction. Now, just like in the aplasia, you can get a temporary increase in the EPSP response, but long-term maintenance of these responses requires protein synthesis. So if you inhibit protein synthesis, you get an initial increase that then decays away over time. Okay? So just like in the aplasia, you have a process of additional continuous stimulation allows calcium influx to modify protein kinases that act through cyclic AMP, that activates protein kinase A, that activates CREB, that causes transcriptional changes, that leads to maintenance of these responses. The details of that are still being worked out. It probably has something to do with maintaining the level of AMPA receptors in the spine head. So there are sort of these 3 separate components to LTP that you should be familiar with. Okay, there's the initial induction of LTP. Okay, there's the expression of LTP, and then there's the maintenance of LTP. So when you're going over these notes, you can kind of break it down into those 3 components. Okay. So I think we'll finish up with this slide and carry on with a little bit of this lecture next time. But I want to kind of point out why we think LTP is a good model for learning and memory. We think it's a good model because everyone basically agrees that you need synaptic change. The synaptic change needs to be dependent on activity. It needs to be specific to which pathways are being active, specifically which connections are being activated. And it needs to have this one, which we're going to talk about on Thursday first, this idea of associativity. Okay, you have to be able to take different types of information and link them together. All right? And there's lots of evidence that shows that if you do various manipulations that affect LTP, you can affect learning and memory. So we'll stop there.